Okay, so we're not taking a quiz today, are we?
Yeah.
We are?
Wait, we don't have to.
We are
No, we are
fine.
fine.
Yeah, no. I thought we took a quiz on Wednesday.
No.
We didn't?
Yeah, we did. Well, I think we were supposed to take a quiz on Monday, but we didn't have school.
Oh, well, I didn't write a quiz.
Oh, that's okay.
Ace for everybody.
You can make it. We'll have one more quiz, but it'll be next Monday.
Can you tell we're all programming?
It'll be on I. It'll be on the special senses.
Oh, just special senses,
The
not
special
the
senses.
first, second class.
The levers and that kind of stuff, no.
What did you talk about today?
And yesterday and last week when we were talking about the I.
So chapter 15. Yeah, I was stressing. So starting.
I was a lot of math. I didn't remember.
Yeah.
And then I didn't remember. I can't remember for my next class either.
So, does that mean we'll be behind on quizzes then?
I generally, if we are behind, then we, I just give everybody 15 points for the last quiz and then just subtract 2 is what I do. So.
Oh, okay.
Is there only one quiz left?
Mhm.
Oh, okay.
So I think we need 7, right?
So it's just.
So I'll just give everybody that 7th one you get full credit for, and then you get subtract
Do those and then.
They get 2 what?
your lowest 2 quiz scores.
Oh, okay.
If we have 7 quizzes.
Oh,
Yeah.
Yeah.
Okay.
Just so you know, you make my week. All week I've been doing, trying to have a week ahead in math now because next week is stock show week. So I'm like trying to get ahead so that when I do my final, like Thursday, then we have this show. Yeah.
So we have one more lab before the lab practical. And I will hand out the review sheet for the final exam during the lab this week, so that you'll have it for a while. Little early.
we turn in our lab notebooks.
You turn in your lab notebooks during the final exam, and I grade them while you're taking the final exam. Yep. So you can use them to study for your final exam. Okay, so we were talking about the eye chambers. And remember that there's tumors in the eye, which are fluids, right? And there's the aqueous, which is watery. And then there's the vitreous, which is gel-like. And so the vitreous tumor is behind the lens, and it makes up a larger compartment. Now, I'm going to show you just a quick video. It's kind of an interesting video that talks about the phenomena of seeing things floating in your eye, and how that actually relates to the vitreous tumor. And then also another phenomenon which you might have experienced when you look at the sky, you can sometimes see flashes of light. And that actually has to do with the movement of blood through the second tunic, the middle tunic, which is referred to as the choroid. So the choroid is the vascular tunic. Okay. So this is just a short little video, talks about those 2 kind of common phenomena in the eyes.
It may look like a tiny worm or a transparent blob. And whenever you try to get a closer look, it disappears, only to reappear as soon as you shift your glance. But don't go rinsing out your eyes. What you are seeing is a common phenomenon known as a floater. The scientific name for these objects is Muscae volitantes, Latin for flying flies, and true to their name, they can be somewhat annoying. But they're not actually bugs or any kind of external objects at all. Rather, they exist inside your eyeball. Floaters may seem to be alive since they move and change shape, but they are not alive. Floaters are tiny objects that cast shadows on the retina, the light-sensitive tissue at the back of your eye. They might be bits of tissue, red blood cells, or clumps of protein. And because they're suspended within the vitreous humor, the gel-like liquid that fills the inside of your eye, floaters drift along with your eye movements and seem to bounce a little when your eye stops. Floaters may be only barely distinguishable most of the time. They become more visible the closer they are to the retina, just as holding your hand closer to a table with an overhead light will result in a more sharply defined shadow. And floaters are particularly noticeable when you are looking at a uniform bright surface, like a blank computer screen, snow, or a clear sky, where the consistency of the background makes them easier to distinguish. The brighter the light is, the more your pupil contracts. This has an effect similar to replacing a large diffuse light fixture with a single overhead light bulb, which also makes the shadow appear clearer. There is another visual phenomenon that looks similar to floaters, but is in fact unrelated. If you've seen tiny dots of light darting about when looking at a bright blue sky, you've experienced what is known as the blue field entoptic phenomenon. In some ways, this is the opposite of seeing floaters. Here, you are not seeing shadows, but little moving windows letting light through to your retina. The windows are actually caused by white blood cells moving through the capillaries along your retina surface. These leukocytes can be so large that they nearly fill a capillary, causing a plasma space to open up in front of them. Because the space and the white blood cells are both more transparent to blue light than the red blood cells normally present in capillaries, we see a moving dot of light wherever this happens, following the paths of your capillaries and moving in time with your pulse. Under ideal viewing conditions, you might even see what looks like a dark tail following the dot. This is the red blood cells that have bunched up behind the leukocyte. Some science museums have an exhibit which consists of a screen of blue light, allowing you to see these blue sky sprites much more clearly than you normally would. While everybody's eyes experience these sort of effects, the number and type vary greatly. In the case of floaters, they often go unnoticed as our brain learns to ignore them. However, abnormally numerous or large floaters that interfere with vision may be a sign of a more serious condition, requiring immediate medical treatment. But the majority of the time, entoptic phenomena, such as floaters and blue sky sprites, are just a gentle reminder that what we think we see depends just as much on our biology and minds as it does on the external world.
Okay, any questions?
The blue skylight one that they're talking about, is that when you see stars?
No, I think that has more to do with like when you see stars, like if you get knocked out, I think that has to do with nervous impulses. So this is specifically when you're looking at the color blue is what they're specifically talking about. So that differs from the aura, the phenomena of seeing the aura when you have a migraine as well. So they don't really understand that aspect of it very well, but sometimes when people get migraines, they start to see light around them and see like stars, you know, that's one of the symptoms of having a migraine.
I thought
Yes.
the reason why you got a migraine, you've seen those, was because it also affected your optic nerve. That's what my neurologist told me, just because it affects your optic nerve.
And it also could be due to constriction of blood flow. Yeah, so it has to do with more with the neurons than it does with what's actually, you know, what you're actually seeing and what, yeah. Yep. Okay. So we're going to talk a little bit more about the eye, and then we're going to move on to some of the other special senses. And so when we're talking about the eye, let me see if I can find my pen, which is missing. Okay, so that's missing. I might have to go steal one from another room. I'll be right back.
I'm waiting, so. Well, I'm working, I need to work on my spaces. And that's the most important thing to do. Not anatomy of it. I gotta really practice.
And then I have to do.
I was giving
Because now you're oh, yeah. What? Wait, so is this your writing?
Okay. So the lens consists of is transparent because this is what allows light to enter the eye. It is also a vascular. So what does that mean? No blood vessels. And so blood vessels would get in the way of transmitting light, and so they are avascular. And they also contain cells that have crystalline proteins. So crystalline proteins. So the lens is able to change shape, which is referred to as accommodation. So does anybody remember the muscle that allows the lens to change shape? What that was called from last? Yes, so the ciliary body. So we can talk about the ciliary body. And when the ciliary body contracts, it actually lets the lens or causes the lens to bulge. So contraction causes the bulging of the lens. So relaxation of this muscle causes the lens to become thin. So this changes where the light is focused. So if we are looking at something close to us, the lens needs to bulge in order to get that image to focus right on the back of the retina. So this would be looking at near objects. So the relaxation of the muscles allowing the lens to flatten out is when we are looking at far objects. So you could put near vision and far vision.
Is that why when they do LASIK for far away, they thin out
The lens,
yeah.
to change its shape. That could be, yeah. So that changing the shape is going to affect your ability to focus the light on the retina. So we have two things that happen with vision. So we have what is called myopia. And so myopia is called nearsightedness or being nearsighted. So what this means is that the lens, instead of focusing the image on the retina, it actually focuses the image in front of the retina. So image is focused in front of the retina. So the opposite of that is hyperopia, which is being far-sighted. And this is where the image is focused behind the retina. So one way that we can change the way that the lens focuses the light is by putting another lens in front. So you can treat myopia, for example, by putting another lens that changes the way that light enters the eye so that the light will focus on the back of the retina. So in your book, they have an image of this. And if you were to take a physics class, oh, I forgot to talk about cataracts. But if you were to take a physics class, you study lenses and you study the way that light is bent as it goes through a lens. And so you'll notice here that light actually gets bent a little bit before it actually reaches the lens. And so what is this called right here, where light enters? That is your cornea, right? And that is actually part of the sclera. It's transparent as well, and it is also avascular. And so you'll notice that the light gets bent a little bit, and then it gets bent even more by the lens. And what we want is that image to be focused on the retina. But you'll notice something else that's really cool about the property of lenses, and that is that actually inverts the image. So all of the images that are coming into your eye are actually getting focused upside down. So your brain actually has to learn to focus those images right side up, right? So everything that you see is upside down, and then your brain takes that image and it turns it right side up so that we see things appropriately. And so if we look at, remember, it is the autonomic nervous system which causes the change in lens shape, because remember that the ciliary body is a smooth muscle which is innervated by the autonomic nervous system. So the parasympathetic activation is what actually causes the lens to bulge in shape. So if that R is really close up, the muscles will contract and cause the bulging of the lens. And then the other one is far vision. Oops, I have a picture of that. Sorry. I don't have a picture of that. So the other one is the far vision, which would be the sympathetic nervous system, which actually causes the relaxation of the ciliary muscle. Okay, so this is a myopic eye. So here we see that the image is focused in front of the retina, and so that is why it is blurry. So that if this is my focal point, then the image that actually gets or hits the retina is going to be out of focus. Sometimes accommodation becomes a problem as people age, because as soon as you hit the age of 40, your lens starts to get really not elastic. So it starts to actually get brittle, what I want to say. Anyway, it stops being able to change shape really easily. And so then you can't see things very well. And so you have to get glasses in order to be able to focus, because the accommodation becomes off. This is just showing how when you put a concave lens in front of that image, then that actually will move the focal point back so that it hits the retina at the right location. And then this is if the eyeball is too short. So this would be a hyperopic eye, right? And then we have to put the lens here so that we can see and we can focus. So this would actually be where the lens is not able to change shape very well. And so you lose the ability to see close up. Okay. So let's go back to the cataracts. So cataracts are specifically due to the clumping of the proteins in the lens. Now you can have cataract surgery where they actually put in a lens. So they take out your lens and they put in a lens. And if it is from somebody else, remember that the lens is avascular, so that it's something that can actually be easily transplantable from one person to another. The causes of this is, for example, you could have a genetic cause, right? So specifically, I know that some breeds of dogs are born with juvenile cataracts, and this is because of inbreeding, right? So like golden retrievers are very prone to having cataracts early on. And so genetic could be a source of this. So you could be predisposed if your grandparent has cataracts. Also, diabetes, mellitus, can give you cataracts. And then excessive exposure to sunlight. Right? So if you go to the eye doctor and they say, oh, it looks like you're starting to get cataracts, they want you essentially to wear eye protection all the time. And it's got to be the eye protection that has the ability to block the UV radiation from the sun. And so when you see eyeglasses, they often say whether they or not they actually protect against UV radiation. So some of them are less protective than others.
Do they do that for kids? Like have them wear glasses?
Right. And so one of the concerns is that like children, say, for example, that hang out at the aquatic center all summer long, you know, they're getting lots of light because not only are you getting light from the sun, but it's also reflecting off the surface of the water. And it's also reflecting off the light colored buildings and, you know, and pavement and stuff. And so it's really bright. And so they recommend that your children wear sunglasses when you go there too, because
my daughter has cataracts, but not like that. Hers are rare. It just looks like.
So do they think it's a genetic?
All over her eyes.
Do they think it has a genetic component?
I don't know. They didn't really say much. They just said that hers were really rare, and the only reason they found them was she would get stomach aches, and then she wouldn't be able to walk. And then they found out it was because she had cataracts.
Oh,
And
interesting.
like altogether, that was what's.
So they didn't tell you anything about using eye protection? Oh,
That's why I was.
yeah.
They
So-
even have goggles now that are.
Yeah, they didn't even tell me because she does swimming. And
Yeah, I would recommend having her wear sunglasses, but you can get goggles that have the UV protection on them too.
her eyes are really bad. None of the goggles they have.
Oh, geez. Yeah, yeah. So this is actually a big cause, right, of cataracts. Okay. Okay, so we're going to talk a little bit about photoreceptors, and photoreceptors have an interesting transduction that is different from all of the other sensory receptors in your body. So they have a unique mechanism of transduction. So remember that transduction is the ability to take environmental stimuli and turn it into an action potential. So most of the time, what happens is that a sensory receptor is when it's not stimulated, it is not producing an action potential. So we'll put most of the time. Okay, when not stimulated and action potential is not produced. So photoreceptors are exactly the opposite. So when we look at the photoreceptors, they produce an action potential when they're not stimulated. So what that means is that they're actually releasing a neurotransmitter in the dark. So they have this process, which is referred to as the dark current. So the dark current refers to sodium ions. So sodium ion channels are open. Sodium ion channels are open. Do you guys have a question?
I'm just a little confused. On the first part, when it says when not stimulated and active, so what is that?
That's all of your other receptors.
Oh.
Sorry, that's a good point. All of your other receptors. So this is how photoreceptors are unique. So they're actually producing an action potential all the time in the dark. So sodium ion channels are open and sodium floods into the sensory structure. Okay, and that causes depolarization. This also causes the neurotransmitter is released, but it is an inhibitory neurotransmitter. And the fact that the sodium ions are flooding in when they're not stimulated by light, this is an example of hyperpolarization. So I'll put when hyperpolarized. So this is hyperpolarization. So this is actually making it so, actually, sorry, this is depolarization. Sorry, depolarization. Oh, okay. So if we look at an example of this, of a diagram, this actually shows the difference between the rods and the cones. And you'll remember that light coming into the retina actually has to pass through the nervous tissue, right, on its way to stimulating the rods and the cones. I'm going to come back to this. Okay, so this is a diagram that you probably should look at. So this is the photoreceptor in the dark, and specifically they're looking at the rod receptors. And so notice that in the dark, sodium is moving in, and then neurotransmitters are continually being released, okay, into the synapse. But it's an inhibitory neurotransmitter. So what this means is that no action potential is actually getting sent to the brain, because this inhibits the release of another neurotransmitter at the next synapse. And so you could write down next to that depolarization is that no action potential is sent to the brain. And remember, it's the optic nerve that transports the action potentials into the visual cortex. Okay. So then if we go to the other extreme, when stimulated, so we'll put when stimulated by light, the photoreceptor becomes hyperpolarized. Sodium ion channels close. And no neurotransmitter is released. This actually, when the no neurotransmitter is released, this actually causes an action potential to be sent to the brain. And I'm not sure why this mechanism is so different than all the other receptors in your body, but it just seems to work opposite, or it does work opposite compared to all the other receptors, like your taste receptors or your mechanoreceptors in your ear, or the odorant receptors in your nose. Inhibitory neurotransmitter is released here, so this does not inhibit the next cell, and so this releases the neurotransmitter, and then an action potential is sent into the brain.
In the diagram, I know you're explaining, that's interesting how like at first when it's in the dark, it depolarizes, and then it says when the neurotransmitter causes it to hyperpolarize. So does it like switch back and forth? Like goes from depolarization to hyperpolarization?
No, it causes the hyperpolarization in the next neuron.
Okay.
So it's in the next neuron. So this causes the lack of the neurotransmitter because it's not inhibitory. So now it is hyperpolarized, right? The photoreceptor is hyperpolarized, but the next neuron that is going to receive becomes depolarized. Yeah, good question. So it's pointing to the next neuron. And that's because the photoreceptors only release inhibitory neurotransmitters. So in the dark, they're releasing the inhibitory neurotransmitter, and then in the light, they stop releasing the inhibitory neurotransmitter.
Are you recording this?
Yes.
Okay. Just so I can have it early.
Let me make sure.
I really hope so.
Yes. Okay. Okay, so let's go back to what is actually causing the change in the photoreceptors. And so one of the really interesting things about our photoreceptors is they have these little organelles that are these discs. And you can see this in the rod. And these discs are actually very similar to the discs that are found in chloroplasts. And chloroplasts are actually in plants, right? So plants absorb energy, they absorb light energy. And we are also capable of absorbing light energy, and our system for doing so is very similar to what is seen in plant cells. But we're obviously not using the light to produce energy like or sugars like the plant is, but we're just absorbing the light so that we can see objects, right? So if we go back to that photopigment, so we have a pigment that absorbs light, and this pigment is called rhodopsin. So we can talk about the photopigment. So instead of chlorophyll, we have rhodopsin. And rhodopsin is actually found in the plasma membrane of these discs that sit, they're specialized organelles, the discs, that sit in the rod and the cone cells. So rhodopsin can actually be converted by light into another set of chemicals. So it can actually be broken down. So if we look at this, I just wanted to write this a little bit bigger. This is retinol plus opsin. So in the light, when light is absorbed, it causes this chemical reaction to occur in this direction. So we would say that retinol and opsin disassociate. So they come apart. So this is called disassociation. Disassociate. Let me make sure I know how to spell that. We could tell it's almost the end of the quarter. I think when the sun comes out like this, it makes my brain stop working so well. Okay, so this is disassociate. There's only one S, sorry. Okay, that's called disassociation. Okay. So in the dark, the retinol and the opsin come back together. Now, retinol is formed from a form of a vitamin. What vitamin do you think retinol is? And you sometimes see this in skin creams. They say, this contains retinol, and what vitamin is that? Nope. 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 C. Vitamin A. Do you know that they named those vitamins in the order that they found them? So that really, and then they tried to rename them, and the A, B, C, D, E, or whatever, all that just, that stuck, right? So it's like not a really good name, because they actually means nothing. Okay, so that is actually vitamin A. And so you actually have to have vitamin A, and one source of vitamin A is like orange vegetables. So like sweet potatoes and carrots and squash, right? That's a source of vitamin A. And so that's why they said that the rabbit eats the carrots to get better vision, right? And so that was kind of a key to try to get children probably to eat their carrots so that they can see better.
I actually know what it means.
Where?
In World War II, they were developing night vision, and so they started the rumor if you eat carrots, vitamin K will increase your vision. So that
Vitamin A, yeah.
Vitamin A, and then the Germans wouldn't know that we were developing night vision.
Oh, interesting.
German told me that.
Yep. So people can actually go blind if they do not get vitamin A in their diet. So vitamin A in developing countries is a really important nutrient that if they don't have it, people can go blind because they can't produce the retinol. And so there was a push in like India to develop a genetically modified golden rice that had vitamin A in it. And so if you're going to eat rice, you can eat the golden version, and that would have the necessary nutrients to produce the pigment. But a lot of people were skeptical of that because why not just give them vegetables or have them grow vegetables that are also have vitamin A rather than genetically modifying rice to make it more nutritious. Yeah. Okay. So this is actually the basis for what we call the dark and light adaptation. So we could talk about dark adaptation. And I don't really like using adaptation in this way because I'm an evolutionary biologist, so that's what I studied when I was in graduate school. So this makes it sound like over generations we're changing, right? But this is actually more acclimation. So you acclimate to the dark. And the reason for this is that when you go from a bright, when you go from a bright light to a dark room, sometimes it's really hard for you to see. And the dark adaptation, so we go from bright to dark. This sometimes causes a few moments of not being able to see, and is called the bleaching reaction. And so this is where it takes time for retinol and opsin to reassociate. Right. So you've all maybe experienced this if you like go into a movie theater during the summer. You walk into the movie theater, you're late, right, and you can't see anything. So if you wait a few moments, what's going to happen is that your eyes are going to adjust, and that adjustment actually has to do with this chemical reaction going this other way, right? So if you wait a few minutes, then your retinol and opsin are going to come back together, and then you're going to be able to see more clearly because you can absorb light, and not all of your retinol and opsin has been bleached, right? And then the light adaptation is where you go from dark to bright. Okay, so you go from dark to bright, and everything looks super bright, and it can be painful. One reason is that your pupil has to adjust, right? So you have constriction of the pupil to prevent excessive light from getting into your eye. But you're going to see things pretty much really colorful when you first come out of a dark room, because that is going to stimulate all of your cones, which all have, they're not sensitive to low levels of light, so they have all of their retinol converted to rhodopsin. So the cones have lots of rhodopsin and things are going to look colorful. That's light adaptation or acclimation.
So what happens when somebody has been in like a dark room for like years? You know, you have those people that have been like captive and then they come out into the light. Is it that their eyes aren't able to, because it seems like they take longer to adjust to the light?
Yeah, I don't know what has happened over years, if that affects, I mean, I don't know. I can't tell you. It also could be that they've been deprived of nutrients too.
Right.
Yeah, so maybe they've been deprived of vitamin A. I don't know. That's a good question. Yeah. And I don't know if I'd want to do an experiment even on an animal to figure out how that would work. Keeping rats in dark, you know. I know that over generations, animals lose their eyes, right, if they're kept in caves. But that would be over many generations. Okay, so if we look at this, this actually is just a diagram showing the shape of the retinol. So in the dark, the retinol has this cis shape, and that you don't need to know what that actually has a organic chemistry meaning, right? So that cis shape, when it absorbs light, it goes and it shows or changes to this trans shape. So the change occurs in the retinol, which then causes it to break free of the opsin. And so the opsin is the protein that's shown here, right? And the retinol is actually shown inside the and that's what causes transduction to occur. Okay. So when we look at the perception of color, remember that this has to do with the cones. And we have red, green, and blue cones. So maybe in cell biology, you studied about color blindness. And the most common form of color blindness is when you lack either the red or the green opsin. So these are different forms of opsin, and they're coded for by different genes. It's different genes. So those genes, interestingly, are on the X chromosome. And that's why they sometimes say that the color blindness is sex-linked, because it is more common in males than it is in females. And so those genes, you could have red-green color blindness. This could be due to the mutation in one of those opsin genes. Okay. So if we look at color, color and visible light is due to the wavelength of photons that are coming into your eyes. And so when we look at color, it has to do with wavelength and frequency. So if we have a wavelength that is very long, so what that means is that the distance between the crests, this would be my wavelength. So generally when we have a long wavelength, then the number of waves in a given amount of time is low. And so we have a long wavelength, excuse me, and a low frequency. So we'll talk about this when we talk about hearing, because sound waves also have different frequencies. So you can hear sounds that have low frequencies versus high frequencies, but the frequencies in the light make us see different colors, right? And so low frequency tends to be here at the red. So this would be long frequency, or excuse me, a long wavelength, low frequency. This would be the red end of the spectrum. And so opsin that is in the red cones is specifically designed to absorb this wavelength of energy. Okay. And then if we have a short wavelength, we generally have a high frequency. Right? So this is a short wavelength, high frequency. And so this would be at the blue end of the spectrum. So notice that in that given period of time, there are more waves of photons coming in. So it has a shorter wavelength. So here they're showing the wavelength of blue, right, versus the wavelength of red. But blue would have a higher frequency than red. That kind of makes sense. It's kind of the opposite. Because we have short, but it's also high. Long, but it's actually low frequency. Okay. So we're going to watch a video that talks about what happens when a color like yellow, how do we see yellow? And so, because we don't have yellow cones. And so we're going to talk about how when we overlap the stimulation of two different cones, how we can see different colors. And this has kind of artistic, is kind of like when you're mixing colors together and what you get. And that is kind of the whole basis for the color wheel, and is the way that we see these.
You might have heard that light is a kind of wave, and that the color of an object is related to the frequency of light waves it reflects. High-frequency light waves look violet, low-frequency light waves look red, and in-between frequencies look yellow, green, orange, and so on. You might call this idea physical color, because it says that color is a physical property of light itself. It's not dependent on human perception. And while this isn't wrong, it isn't quite the whole story either. For instance, you might have seen this picture before. As you can see, the region where the red and green lights overlap is yellow. When you think about it, this is pretty weird. Because light is a wave, two different frequencies shouldn't interact with each other at all. They should just coexist, like singers singing in harmony. So in this yellow-looking region, two different kinds of light waves are present. One with a red frequency and one with a green frequency. There's no yellow light present at all. So how come this region, where the red and green lights mix, looks yellow to us? To understand this, you have to understand a little bit about biology, in particular about how humans see color. Light perception happens in a paper-thin layer of cells called the retina that covers the back of your eyeball. In the retina, there are two different types of light-detecting cells, rods and cones. The rods are used for seeing in low light conditions, and there's only one kind of those. The cones, however, are a different story. There are three kinds of cone cells that roughly correspond to the colors red, green, and blue. When you see a color, each cone sends its own distinct signal to your brain. For example, suppose that yellow light, that is real yellow light with a yellow frequency, is shining on your eye. You don't have a cone specifically for detecting yellow, but yellow is kind of close to green and also kind of close to red. So both the red and green cones get activated, and each sends a signal to your brain saying so. Of course, there's another way to activate the red cones and the green cones simultaneously, if both red light and green light are present at the same time. The point is, your brain receives the same signal regardless of whether you see light that has the yellow frequency or light that is a mixture of the green and red frequencies. That's why, for light, red plus green equals yellow. And how come you can't detect colors when it's dark? Well, the rod cells in your retina take over in low light conditions. You only have one kind of rod cell, and so there's only one type of signal that can get sent to your brain, light or no light. Having only one kind of light detector doesn't leave any room for seeing color. There are infinitely many different physical colors, but because we only have 3 kinds of cones, the brain can be tricked into thinking it's seeing any color by carefully adding together the right combination of just 3 colors: red, green, and blue. This property of human vision is really useful in the real world. For example, TV manufacturing. Instead of having to put infinitely many colors in your TV set to simulate the real world, TV manufacturers only have to put 3: red, green, and blue, which is lucky for them, really.
Okay, so the idea there is that we can see the different colors besides red, green, and blue because of the overlap. So that in some cases, if you're talking about yellow, that would stimulate both the red receptors and the green receptors. And so then the overlap sends a combination of action potentials to your cerebral cortex, which makes you consciously perceive the color yellow. So even though we have only 3 different cones, we can see a variety of colors because of this overlap. And so this overlap is actually shown in this diagram. So notice the green cones can actually see here. So they would be stimulated right here within this wavelength. And the red cones are stimulated within this wavelength. And when they're stimulated, the retinol changes shape, right? And then an action potential is generated and it goes via your optic nerve into your visual cortex where you perceive the color. Okay, so we're going to move on to another type of sensation, which is taste. And so the taste receptors are called gustatory receptors. And you should remember that word because we did talk about the gustatory cortex, which is responsible for taste. And these gustatory receptors are primarily located in the taste buds on the tongue. Now, weirdly, there are some taste buds on your cheeks. There's some evidence that there's some taste buds in the back of your throat, like on your uvula, which is that thing that hangs down in the middle. But really, most of the taste buds are located on the tongue region. And the gustatory receptors sit inside of these taste buds, and they have on them receptors that are able to detect the 5 different taste sensations. Okay, so we have 5 different receptors. Okay, so we have salty, sweet, sour, bitter, and umami. Umami. I think that's Japanese. And so salty gives us a sensation of sodium, like how much sodium are other minerals that our food has in it. So if we excessively sweat, we can actually lose a lot of salt. And so we can crave salty foods in order to recover that. Sweet gives us information about caloric content, energy content. So if we don't eat a lot and we are really, really hungry, we need nutrients, oftentimes the first thing that we'll go to is something that is sweet. Sour has to do with what? Acidity. which is pH, right? But vitamin C is also sour. So citrus, we need vitamin C in our diet. It's not something that we can make. And so we have to take in citrus, vitamin C, in order to be able to survive. And then bitter oftentimes has to do with toxins that are in vegetables. So there's some vegetables that are more bitter than others. But we can learn to like bitter tastes, like the bitterness of dark chocolate or the bitterness of coffee, right? And then umami is what? Savory, right? And so that's protein. And so I think I mentioned when we, seems like have we talked about these sensations this quarter?
No, I think it was
233, yeah. So umami is one that the food industry has utilized to their benefit because it would include foods that have MSG, monosodium glutamate, added to it. That is a flavoring. And so like those ramen noodles, right? You are tricked into thinking that those are full of protein because they add MSG, which makes them taste savory. But you eat them and you don't get anything but really carbohydrates and salts, right? And so they don't have any protein content to them, but it kind of tricks your body into thinking they have protein in it. Same thing with Doritos. Doritos always, to me, taste like they have protein in them, but they're not, right? Okay. So if we look at where these gustatory receptors are located, okay, so this is my taste bud, right? And then in the taste bud, we have these receptors. And the receptors have these little hairs on them, and they actually bind to the chemicals. And then ultimately, the binding of a chemical causes depolarization. So binding of different chemicals ultimately leads to the depolarization. So this is the normal mechanism of transduction, right? Unlike the photoreceptors, which are depolarized even when they're not absorbing the stimulus. And this mechanism of transduction is a little bit different for each of the receptors. So sometimes they bind directly and cause sodium ion channels to open. Sometimes they use different ions, but that ultimately leads to depolarization. So remember that this sensory information is transduced into an action potential, and it gets perceived, right, in the gustatory cortex. So if we were to look at the nerves that innervate, the cranial nerves that innervate the tongue, we have the glossopharyngeal. So glosso means tongue. And then we also have the facial nerve, which also innervates the tongue. And then the vagus nerve, which is the epiglottis. And supposedly the epiglottis, which is kind of, it's way down in your throat, that has some taste sensation as well. So that all gets sent, and remember that it goes up through the thalamus, which is the major relay center, and then that action potential gets sent to the gustatory cortex, where you consciously perceive that stimuli. So in the past, there's been this idea that the taste buds could be mapped. Like, for example, most people could taste bitter a little bit better in the back of the tongue versus the sides of the tongue. But that mapping of the taste buds seems to have been disproved in that they generally occur all over. So you can taste bitter and sweet and salty and sour all over your tongue, not just in a particular region. So if you did that in my grade school where you tried to map the tongue, that was a really common experiment, but they don't think that that is true.
Does that also differ depending on the person, or is that?
Yes, and so one of the really interesting things about taste buds is that there truly are people that are super tasters. So a super taster is a person that has a lot more taste buds than everybody else. And so they can perceive taste a lot better than the normal person. And so they think that people that are like food or wine or other connoisseurs and can write about it, extensively about the different tastes, They just seem to have more receptors than average. And it's interesting, females seem to be better at tasting than males. So, yeah, so, although that is not true with my husband. He is much better tasted than me. So that's, in general, females tend to be.
Is that also, kind of backtracking a little bit, is that also the same with color? Like, her and I looking at this, do we generally see the same color, or most people take it in?
You generally do see the same color unless you have a mutation that prevents you from distinguishing red and yeah. Okay, so that is the taste buds, and these are chemicals in solution. So you might want to write that down. Chemicals in solution. So water, right? So they have to be mixed in solution in order for you to be able to perceive it. And this is different than smell. So these are airborne chemicals. And these are called odorants. So rather than just having 5 major receptors, they have discovered when they did the Human Genome Project that we have 400 smell genes. And so supposedly these smell genes code for different receptors that bind different chemicals.
That way some people might think lavender smells like really great and other people don't.
What? Think what smells good?
Like lavender.
Oh,
Some person might
lavender.
think lavender smells really good, but like my husband thinks it smells horrible. Is that like because of the gene receptor for smelling it differently?
No, I actually, well, maybe. It could be that his perception of it is unpleasant. Maybe, I'm not sure. I mean, there could be a variety of things. Sometimes we learn to not like a smell because of past traumatic events, right? And they've actually discovered that that can be passed down weirdly from one generation to the next, because this is a crazy experiment, but they took these mice and they tortured them and gave them the smell of cherries. And then they took the mice's offspring, the offspring, the babies had never been tortured in the presence of cherry, the smell of cherry, but they exposed them to cherry and they had the same fear response to that smell as the parents did. So they think that you could pass down memories of bad stimuli from parent to offspring. It's crazy. Like, it gives me goosebumps every time I think about that, because it's like, oh, why do I like this or not like that? Maybe that has something to do with my ancestors, right? Something happened to my ancestor, right? Okay, so they code for different receptors, and they think that these can combine, right? So odorants overlap, right? Odorants overlap. So smell is complex. So it's much more complex than taste. And oftentimes what we perceive as we say, oh, that tastes good, it actually smells good to us. Right? So like the smell of cooking onions, for example, is a nice smell, right? That most of us like. But that is not just one odorant, but it's a multitude of odorants, and so that gives you the smell.
So what's the association with smoking? Because I know that if I'm taking bad medicine, if I plug my nose, I can't taste it. And that's what I've been taught to do.
Right, and so what I'm saying is that sometimes what you think is taste is actually smell.
Right.
So like if you're taking that cherry medicine, right, that icky cherry stuff, I don't know who came up with that.
Which one, the NyQuil?
Cherry, just anything but cherry cough medicine.
I think it's great. I drink NyQuil.
Anyway, so if you do plug your nose, well, the only thing you're gonna taste is sweet. The whole cherry sensation comes from smell. You're also gonna taste bitter because the chemicals in it are bitter. But they're trying to cover the bitter with sweetness. Right?
They fail.
So bad medicine is sweet, bitter, right? The whole smell is the, grape versus cherry.
Yeah, I don't do orange flavor. Orange, that's my no-no.
Okay. So the other thing about these receptors is that they have a short lifespan. So they're actually replaced every 30 to 60 days. So you can damage your receptors by taking, like smelling a bad volatile chemical, like maybe ammonia, right? And you might not be able to smell right for a few weeks, but eventually those receptors die and they're regenerated and they're replaced. The problem is that as we get older, they're replaced less frequency. And so people that are older stop losing their sense of smell. And because they lose their sense of smell, they also lose their sense of taste, because that's important. And so older people will stop eating because things just do not smell good, right? So they have no appetite because they're not smelling the good smells that would normally stimulate the digestive process.
So like when you get sick with a cold or something and you don't really taste or smell anything, is that because you're sick, or is that like your body is just kind of like, well?
That is because you're sick. And so you produce mucus that prevents odorants from getting up into and binding to your receptors.
So then because your nose is plugged, that's why you can't.
Yeah. Yep. Okay, so if we look at this diagram, so these receptors are located way far up in your nasal cavity. So this is like way far up where your nose sometimes bleeds if it dries out, because the lining of your nasal cavity is actually just a single layer of epithelial tissue. So notice that this is located at the olfactory bulb. So the receptors dangle down. They're kind of in this mucous membrane, and they bind the chemicals, and then it gets sent directly to the olfactory bulb. So remember that the olfactory bulb, so we'll say the receptors send, I'll just put action potential AP, to the olfactory bulb. Okay. This is not conscious, but connected to the limbic system. The limbic system is that emotional response, right? So smells can sometimes initiate an emotional reflex without us actually consciously perceiving them. So that olfactory bulb they think that this is one of the most ancient of our sensations, and oftentimes this sensation is not consciously perceived. So for it to be consciously perceived, it has to go to the olfactory tract. Right? It has to go through the thalamus. And then it goes to the olfactory cortex. And this is where we have conscious perception. And smell is really interesting because it's very difficult to talk about. Like even taste is easier to talk about it than smell. So if I was to ask you, like, what does somebody smell like? It would be very hard for you to consciously think about it and make comparisons between individuals. Like, how do these two people different smell? Right? It's not something that we put into words. We don't have the language for it. So, or how does this room smell, right? But remember that smells can be associated with memories. And so oftentimes when you're asking somebody, what do they smell like? It could be like a memory of a smell that you've had in the past. So a good example is every time, you go to the grade schools and they have that powdery soap, everybody knows what that powdery soap smells like. Oh, yeah.
You must have had a good child.
Or like the time of year. Like you wake up one day and you're like, oh, it smells like fall.
Yeah.
It's like overnight.
Okay, so the next thing that we're going to talk about is hearing. So we're going to stop here for today, and we'll talk about hearing, and then we're moving to the endocrine system. That's the very last topic.
It probably makes it to it. That's okay. So my daughter's here is gonna do